It is absolutely my pleasure to introduce today's Pioneers in Biomedical Research Seminar Series speaker, Dr. Ondine Cleaver. So a little bit by way of background, Dr. Cleaver's academic career began as an undergraduate, so we're going to go way back, Ondine. So hold on, here we go. Hopefully I get all the details right. Oh, yes, high school. Oh, I should have, it's not in her CV, so it's not my area here. So, okay, so she did her undergraduate at UT in University of Texas in Austin. She got a BA in history and a BS in molecular biology. So the word polymath came to mind for any of us who might know what that means, but a jack of all trades, if you will. Followed by, so to continue that, her graduate education in zoology, right? With a focus in molecular developmental biology and neovascularization, also at UT Austin with Dr. Paul Krieg. She went on to a postdoctoral fellowship at a little place we like to call Harvard University. So it's very prestigious there with Dr. Doug Melton. It was here during these years that Dr. Cleaver contributed some numerous impactful publications. One of my favorite being how she elucidated how the vasculature facilitates organ development, which is just one of the themes that emerged from that work that's gone on and held true for many years. She did that specifically looking at paracrine factors from blood vessels in the pancreas that drive endocrine progenitor cell expansion and islet cell differentiation. So really some seminal work there. Then Dr. Cleaver began as an assistant professor at the University of Texas Southwestern Medical Center in Dallas, so not straying too far from Texas, going back to home, I guess, in the Department of Molecular Biology, while also assuming the role of co-chair in the Genetics and Development Graduate Program. She continued on the promotion path to associate professor with tenure in 2012, and then full professor in 2018, all again, which at UT Southwestern. And she is also the chair of the Genetics and Development Graduate Program. And her role as chair in the graduate program, she was discussing this last night, is not surprising given on her CV. Ondine's CV is just packed with her passion and talent for mentoring and teaching, reflected in her numerous classes, panels, and activities that Ondine has led over the years to raise up future scientists, including trainees from her own lab. In fact, she guest lectured in one of my classes just fairly recently, and my students recall her lecture with much fondness, telling me that was their favorite lecture of the whole semester, including compared to my own. So Ondine's challenging me to raise my game up here in terms of teaching, because she definitely gave the best lecture in my class. Mason would agree. Dr. Cleaver has won numerous awards. I sort of lost track, so I just said numerous because I was like, I can't list all of these. She's an integral member of a wide range of advisory boards, organizing committees, review panels for funding agencies, organizer of conferences, including some highly regarded Gordon conferences and International Vascular Biology meeting. Ondine was recently the president of the North American Vascular Biology Organization, affectionately known as NAVBO, which included, at this more recent meeting in California, an amazing dance party that featured Ondine and myself on the NAVBO, I guess at that point, Twitter feed, sharing some epic dance moves with trainees. So, showing them how a good time also. I can share that link later if people are interested. Maybe not, maybe not. Anthony? No? Anthony, no? Okay, all right. All right, then just yes, fine. Only Jess. All right. So Dr. Cleaver has published over 90 book chapters, reviews, and journal articles. Oh my gosh, it's exhausting. I know, right? You're more prolific than even you thought. Top tier journals such as Science, Nature, and Developmental Cell. She's been prolific in attracting funding as well, being a PI or co-PI on 6 NIH-supported grants currently, as well as a Leduc Research Foundation. Okay, so I could certainly continue to share Dr. Cleaver's wide range of accolades and successes, but I wanted to offer other reasons why Dr. Cleaver is one of the scientists whom I hold in such high regard, wanting to model my own career after hers. I met Ondine when I was a postdoc at the University of North Carolina in Chapel Hill, and it was at a dinner at a conference one evening where Ondine took such a genuine interest in our projects and careers as trainees, sharing her own experiences and offering encouragement. I mean, she was just doing it just before this talk even. She says, you don't even realize you do it, but you just support everyone you're around. It's an amazing feature, personality trait, excuse me. And she shared, that encouragement and offering guidance for our careers. So, and then I've also heard Ondine give many research presentations over the years, and I've been continually struck at how she presents and discusses her data, giving the sense of being guided by curiosity and wonder rather than self-promotion and agenda. I've had the pleasure of interacting with Ondine in several other capacities over the years, and she has always been a source of encouragement, honest feedback, and a kindred spirit and pursuing our shared fascination with the vascular system. So there's so much more I could say, but I wanted to close with the introduction by saying thank you for making the effort to be here today. I know you travel quite a bit, so adding one more travel I'm sure is a bit exhausting, but we appreciate you being here to discuss your exciting work exploring the mysteries of tube formation, which is a fundamental process in the origins of life on our planet. So without further ado, Dean, I'd like to welcome you.
Yeah. Thank you so much. Make me cry. I don't think I've ever had such a wonderful introduction. So thank you so much. You did. And thank you all, those of you who've been meeting with me, dinner last night. I mean, it's wonderful to be here. Wow, you have an amazing group, interactive, friendly, doing really beautiful research. So I've really enjoyed being here. So thank you for being here. So today I'm going to tell you about work that I'm interested in that really started when I was a graduate student. And you mentioned that I was a history major. I was, and I took my dad forced me to take science and math and everything. And I took a molecular biology class that was taught by a developmental biologist. And I was just immediately struck. I was like, this is what I want to do for the rest of my life. And I continue to just do work that I find fun and interesting. And I hope that all of you students follow that. What you love to do is what you're going to be really good at. So don't give up. It can be tough, but it's fun. So what I'm going to tell you about is how to build and maintain blood vessels. This is what I started doing as a graduate student, and which has really kind of stuck with me and has expanded into other tissues and other organs. But it's essentially asking the simple question, how does a cell in the developing embryo make a decision at some point to, one, become an endothelial cell, which is a basic building block of blood vessels, and two, know where to go and who to associate with and how to maintain that structure once you've built that structure. And that has really sort of guided that. These questions have guided me through my career. So it's recently took kind of an interesting sort of sidebar. I've been pulled into an effort, a biomedical engineering effort that maybe would be interest to a lot of you, in terms of trying to build organs in a dish for replacement therapies. And this interest sort of started when I was in Doug Melton's lab, where we were really looking to make islets to be able to transplant into diabetic patients, but has recently shifted to, for instance, developing some kidneys based on approaches that I'll discuss more towards the end of this talk. But the idea is, can we make these tissues? They are made properly in the embryo. So why can't we learn all those rules and borrow that and apply it ourselves? Lots of people are waiting for organs. Every day people are dying, waiting for those organs to be made. And so the question really is it possible to make them? So this is going to be the outline of my talk. It's going to be three major parts. I'm going to talk about the building of blood vessels themselves. How does that happen? What are the rules? What are the requirements between cells that need to be just right to make a functional blood vessel? And that is going to be this part two of this talk here is going to be a focus on a gene that's only expressed in blood vessels and really important for building blood vessels. Then I'm going to basically tell you the same story, kind of, but it's going to be a different gene, similar but different gene. And it's going to be required instead for pancreas tubes to be formed. But what I'm going to try and do is show you how the same molecular machinery can be required between two very different tissues to make the same outcome, which is a biological tube. And then in the end, what I'm going to do is tell you, okay, once you've built a tube, and I'm going to go back to blood vessels, how do you now maintain it? What are the rules for maintaining blood vessels? So it's going to be about building, and it's going to be about maintaining. And it's going to feel a little bit like whiplash because there's going to be a lot of different organs. There's going to be the blood vessels, the pancreas, and the kidney, but it's all kind of the same story. So I'll try to bring you back up to that level and remind you why I'm jumping around a little bit. So just to start off about building organs, and it's just kind of introduce a little bit about my interest. What can we do learning from the embryo? This is this effort that I mentioned about trying to build organs in a dish. I was pulled into an NIH consortium effort called Rebuilding a Kidney. And in this effort, we have everything from basic scientists, cell biologists, bioinformaticians, clinicians, bioengineers. And what we're trying to do is simply to say, what's the best way to put a very complicated organ together? We know what all the cell types are, and so can we just put them together and then have them function? So there's lots of different ways that people are approaching this. And I would say that we've had this effort for the past 7 years, and we've basically just knocked off all the ways that you don't do it. And so one of the efforts that went fairly quickly was this one that I'm showing you here, which is essentially melting off all the cells, decellularizing an organ, and then trying to get the cells back into that organ to go to the right places and remake the organ for transplantation. So let's say you take a kidney from, let's say, one patient, but those cells are going to have an immune reaction, and so you decellularize it, you add the new patient's cells back in, and that way it makes it transplantable. We've tried that, and it's been really tough. The cells don't know where to go, even though there's an extracellular matrix scaffold left behind. Lots of other methods have been used, including regeneration approaches, but the one that we've really narrowed on have been this organoid approach. And like I mentioned, I'm going to talk about that mostly at the end of the talk. But why I want to bring this up is that it's a system where these cells start off very naive, like they do in the embryo, and then you give them some signals, and then they start to assemble sort of spontaneously, same that they do in the embryo. So my philosophy has really been that we can learn an enormous amount about how to build tissues by just going back to the embryo. This is a quote that was on the wall when I was a graduate student by an old embryologist, Victor Hamburger. Our real teacher has been and still is the embryo, who's incidentally the only teacher who's always right. It's an amazing thing that all of us here look identical, basically. We have 2 eyes, a heart, cardiovascular system, muscle. How does that happen right every time? Well, there's rules for it. And there's not a million rules. There's probably in the thousands of rules. Can we learn what all those rules are of how cells grow, proliferate, interact with each other, induce each other? And so these are the things that I'm interested in, which is really called morphogenesis and cell-cell interactions. And so everything really depends when you build a tissue on these basic fundamental behaviors that I'm going to touch upon when I tell you about the work that we've been doing in the lab. So cell shape, especially cell adhesion, are 2 of the rules that we're going to focus on. So what about tubes? You probably, when you were trying to decide to come to this talk, you were like, oh, does that sound interesting? What's a tube? I was reminded last night at dinner that actually one of the first multicellular organisms in fossil record, and so John was telling me last night, turns out to be a little tube, these little animals that were really just epithelial tubes. You have an in, you have an out, it eats, it gets rid of things, and that's it, they live. Tubes are actually really important in all of you as well. A lot of our organs are actually made up of tubes. And so here I'm just showing you a couple, but mammary glands, lungs, salivary glands. And we have studied in our lab all 3 of these tubes down below here, blood vessels, pancreas, and the kidney. And this is what is so fun to me, is that there have been similar rules between these, what seems like wildly different tissues, but in fact, there's some similarities. So today I'm going to focus mostly on 2 of these, which is going to be the endothelial cells, which line all the blood vessels, and the epithelial cells, which line pancreatic tubules. And I'm going to tell you how there's similar molecular machinery between these 2. So just to describe what you're looking at here in this slide, on the left in this panel here is a very early embryo, and you're going to see a lot of these images here. This is a close-up of a mouse embryo, and you can't really see the neural folds, which is probably of interest to many of the neuroscientists here. What you are seeing in green is a reporter for early endothelial cells. And this is the yolk sac, so there's lots and lots of blood vessels. This is the early heart, it's just a little bag at that point. And these are the vessels that I really spent 10 years studying when I first opened my lab, which are the dorsal aorta. And there's 2 of them, and they fuse together and they form the dorsal aorta that's in all of us and is the main conduit from the heart to the periphery. But all blood vessels in the embryo are lined by these very skinny cells called endothelial cells that are associated with each other via junctions. They know where the inside is, which is where the blood is pumping, and they know where the outside is, which is where they associate with lots of other cells, including smooth muscle cells. So if you've ever seen a large blood vessel, there's a really encasing of smooth muscle that's not shown here. On the right-hand side are the pancreatic tubules, and this is probably an image you've never seen before, but the pancreas is really weird, and I'm going to take you through it, and it's really fun to study. But it is a plexus of tubes. It's a three-dimensional net of all kinds of intersecting tubes, and what you're looking at here is actually a marker that is on the inside of the tube. So it's as if I've painted the inside with a paint, but you're not seeing the epithelium. So it's just sort of outlining these lumens within the pancreas. And we're going to talk about how they form. But if you take a cross section through all of them, I've just described the endothelial cells. But if you now compare to an epithelial tube, and a lot of them kind of look like this, the epithelial cells are very thick, cuboidal or columnar. But they're similar in a way. They're associated with each other via junctional surfaces. So they touch each other. They know where this apical or luminal surface is, and they also have a basal surface on the outside, which is associated with ECM and with other cells on the outside. So these are just the basic structures that I want to take you through. So I'm going to start off by going through the blood vessel work. And I'm going to focus on a gene that I discovered a long time ago and basically opened my lab up. All of my graduate students worked on this originally and it was unknown at the time. And so it was really fun trying to figure out what it was doing. But the upshot of what it does is that it controls GTPase activity. And so just to remind you guys, because I had to be reminded when I found this, GTPases are these small catalytic molecules that go back and forth between an active and an inactive state, depending if they have GTP or GDP, and they help catalyze reaction with other molecules. But when I first found this, I was really depressed. I was like, oh, I don't want to work in the cytoplasm. I don't want to work on GTPases. It's too complicated. But I've ended up having a lot of fun to do it. So to sort of give you the background of where we are studying, this is our playground here. This is an early mouse embryo. And do I have a pointer? Is this, let's see, maybe it's, okay, yeah, on and there. That's much better. Okay, thank you. So in each one of these panels, what you're looking at is an embryo. And this is time. And so you're going from an earlier stage to a later stage. And we mark time by somite. So somites are bundles of muscle, and the more of them you get as the embryo develops. So at the one somite stage, this is what a mouse embryo looks like. It has no other organs except for perhaps these neural folds just starting to form. This is, again, this reporter that shows you where the endothelial cells are. So the yolk sac is full of blood vessels, but the embryo itself is this darker little pizza slice over here. And what you're looking at here are these cells that are emerging in the mesoderm as the embryo is forming. These are the precursors to the endothelial cells called angioblasts. They're like little amoeba. They're migratory. They're trying to work their way through the mesoderm and find each other, which they eventually do. And when they find each other, they adhere to each other, and they're now going to end up forming these little linear aggregates, which are called cords. And then these cords will undergo what I find to be this amazing transformation where they actually open up a lumen on the inside. So this is the process that we're going to be discussing today. So I'm just going to show you this again in kind of a different view. This is the whole mount view, the angioblasts, the cord, and now the tube. If you cut this on the transverse section, you can see these individual cells migrating over the endoderm. You can see them joining together. I've just kind of false colored the junctions. They have to make adhesions with each other. And then during tubulogenesis, they now have to clear that apical membrane. So the whole point of this first third of the talk is going to be, how does that happen? How did those junctions move out of the way? It's a really critical process. Yes, please. So that's exactly right. There's lots of mesoderm that is not somitic. In fact, probably the majority of it, lateral plate mesoderm, and yeah, exactly right. Yeah. Yeah, please.
Does this process of tubulogenesis proceed as a wave?
Absolutely.
Or is it just, oh my
I took out a slide because I didn't want to complicate it, but you're absolutely right. So it opens up actually at the heart, and then it sort of doesn't zipper, opens up from anterior to posterior. And the heart, I believe, actually helps in that opening of the first initial vessels because it pumps. There's no blood at the time when it starts to pump, but you can see it really well in the zebrafish, actually, where the intersemitics, the pumping opens up those vessels. So yes, very important question as to how the circulatory system eventually forms. But so down below here is just a cartoon of representing what I've shown you. So when I first started my faculty position, one of the things that I carried with me from my postdoc lab was long lists of genes that were expressed in various portions of the embryo. We were looking for something different. We were looking for the signal from blood vessels to early beta cells. But what I was able to take with me was just, okay, what's expressed in the endothelial cells? And so I started off in the first couple of months of my faculty position just doing in situ hybridizations. And in situ hybridization just shows you where a certain gene's expressed. And so what you're looking at here is just a result of that screen. And so it's a very complicated, lots of panels here, but what you're looking at is day 8.5 or 9.5 mouse embryos, which I've done in situ hybridization on. Here's a face view showing you those 2 dorsal aortae on each one of those embryos. Here's just a section showing you the dorsal aorta in cross section. And then here's a day later just showing you the vasculature as it's elaborated and it's become more complicated. So, but what I want you to get from this is just look at these names of these genes that came up from the screen. These were just terrible to see. I was just like, oh my gosh, Rho GTPases and RAP. And why are there so many of these GTPase associated genes? As a developmental biologist, I wanted to work on transcription factors and receptors or growth factors. I did not want to work in the cytoplasm because I didn't know it very well. But when biology tells you something, you just kind of have to follow it and you kind of have to learn it. So I started learning it. But there was so lots and lots of these effectors. So what did that mean about, you know, how these cells were essentially using these molecular pathways to start building these early blood vessels? These were expressed really strongly in them. Some of them stuck around, some of them went away, but it was clear that GTPase regulation was going to be important. So I actually asked my chairman, I said, you know, how should I pick? I have all these beautiful genes. And he said, listen, he says, any gene you knock out, 70% chance you'll have a phenotype. And I went after the top gene on the list, RAS-IP1, there was nothing known about it, and knocked it out, and it ended up having a phenotype. But here was the very brave soul who was at first a technician in the lab, then became a graduate student with me, who ended up tackling this project. And so what he did was to establish that one, this gene was beautifully and only expressed within the endothelial cells, not in other cell types. It was conserved among different organisms, frog, fish, mouse. It was a molecule that actually there was very little known about it. There was only one person that had really worked on it who was a GTPase person. She was a RAS person. She did a yeast 2 hybrid screen and she got it. She just found it and she published on it, but they didn't really know what it did other than it was associated with all these GTPases. And we ended up becoming good friends and she ended up giving me lots of good information. This is what it looks like. It is basically a scaffolding protein. It hangs out in the cytoplasm. It moves around. We know that from some experiments I won't show you. It has a RAS association domain or a GTPase association domain, protein-protein. And then an interesting domain that's going to become relevant is called a dilute domain, and it's implicated with moving cargo around within the cell. So Kerr was like, okay, this looks really interesting. We're going to have to learn about GTPases. There was no known function in the literature. Therefore, this seems fun. So he started off just doing simple experiments in vitro up here and in vivo to just ask, might it be important? If he knocked out this gene with siRNAs just in endothelial cells that were plated on matrigel, he found that something happened. The endothelial cells didn't behave as they should. They should form this little plexus, this is the way they do normally, both in a dish and also in the embryo. They failed to do that. They rounded up. If we knocked it out in a frog, which was what I did as a graduate student, was work on frogs, you could see that on the uninjected side, blood vessels were fine, and on the injected side, blood vessels were affected in all kinds of different ways. Some weren't there, some were disrupted. So then I did what my boss, Eric Olson, suggested, knock it out in mouse, and it was wonderful that there was a beautiful phenotype. At first, we thought there was no blood vessels, but then as we looked further into it, there were blood vessels, but they weren't functioning properly, and these embryos died very quickly of cardiovascular defects. So this was exciting, and had it been 1994, we could have published it in Science, and it would have been great. But you all, as science proceeds, we got to figure out mechanism, and this is what we have dived into within the last 20 years and really figured out what RAS-IP1 is doing. But the upshot of this was, the phenotype, was that it didn't open up lumens properly. And so normally, endothelial cells, what they do in this early embryo is to line up in these cords, and then what they do is to have to move junctions out of the way to open up this lumen. And they open up a lumen, and then they start to proliferate. There starts to be more, the diameter of the vessel increases, and they also change shape. They start off very cuboidal. If you remember some of the images I showed you, these angioblasts are very rounded. But when they end up in a blood vessel, when they differentiate into an endothelial cell, I always liken it to an egg. An angioblast is an egg, and if you crack it and if you cook it over easy, that's what differentiation looks like for an endothelial cell, and the yolk is the nucleus, but it becomes very, very thin. And so what we noticed in our RAS-IP1 mutants was that the cell shape was definitely not doing this kind of thinning. It remained very cuboidal and very rounded. We also noticed that it lost its adhesions to this outside mesoderm. And you were asking about the outside mesoderm. This is called sclerotome here, and the sclerotome has morphogenetic power. It opens up this lumen regardless of whether or not endothelial cells are there, but these endothelial cells lose adhesion to it. So we showed this in this paper. It was a long time ago, so I don't want to go into details, but adhesions to this outside world was lost. And then there was also the question of what is going on in the association between these endothelial cells. Here, what you can see is that they've opened up and they've cleared junctions. Here you can see that they really remain adhered, and we're going to look at that in a lot more detail. So this is looking at it a little bit more closely. Normally, endothelial cells will do the following. They will, let's say you have 3 endothelial cells. They start opening up this lumen. Here's the apical surface. And what they've done is that they've moved aside junctions. And so Z01 is a tight junction. And they've moved Z01 out of the way. They've opened up this little lumen here. And so you have this kind of polarized cell. You have junctional region, you have apical region, and then you have the basal region around the cells. Now in the RAS-IP1, which was what was interesting, was that you kind of had semi-polarity was okay because you had this apical marker. This is podocalyxin. It's a sialomucin, which gets put on the apical surface, and it is normally localized at the apical region. But when you start taking a look at these junctions, they're no longer brought to the periphery of the lumen, and instead they stitch it together. So it's as if instead of opening up this cohesive, this continuous lumen, you now just have all these little sticky points in between, sort of stitching it up. So that was the main phenotype that we had to deal with. And so at this point, I got a new guy in the lab, David Berry, who joined in. And that was the first thing he asked when I was kind of in my office describing what the projects were. And I said, this is the phenotype that we get. And he goes, why? Why are these junctions not moving out of the way? And I said, I have no idea why they're not moving out of the way. Normally, they would be cleared, but in our RAS-IP mutant, they're not cleared. And so this is the work that he took on after Kerr's work to ask that question, what are the forces that are really important for moving those junctions out of the way? Okay, so the answer really came in a nugget of information that was done by Kerr, but Kerr had done a mass spec to just simply ask the question, who binds to RAS-IP1? Maybe this will give us some clues as to what RAS-IP1 is doing. And when he did that, he came up with 2 really interesting proteins that caught our eye. And one of them was this molecule non-muscle myosin chain 2A. And one was a molecule called RGAP, which is a gap for a row, something that will inactivate this GTPase, pretty well-known GTPase row. And so why is that interesting? So I just kind of put the complex together here, and I just want to tell you a little bit more about these molecules. This non-muscle myosin is sort of like the muscle of the cell itself. So it has these long chains here. It also has these heavy chains that bind to the cytoskeleton. And essentially, when this molecule is activated, it constricts. So that's its job. Now, RAS-IP1 was not known to be associated with this before as a new molecule, but here it comes in, with these reciprocal pull downs, we see that this complex is really important, and it's carrying with it this gap. So why is that kind of interesting? Well, what's known about non-muscle myosin is that it is activated by Rho. So here's a Rho inactivator, and here's a Rho activatable molecule. So how does it get activated? These little purple light chains here, if they are phosphorylated, they activate this molecule. So here's a molecule called Rho activated kinase. And if it activates this light chain, then it activates the whole molecule. So who turns on Rho kinase? Well, turns off its Rho. And then who turns off Rho? It turns out there's a whole series of these gaps that turn off Rho. So, we had this complex and we had to decide, okay, how is this acting during lumen formation? So we started simply asking the question, where are these complex members? Where are they located, and how might they be playing a role in lumen formation? So what I'm just showing you here is that these molecules are active right on top of the junctions. So I am showing you here an activated myosin light chain. That's that little purple component that I showed you previously here. So that's that component here. And when you do a stain for phosphorylated myosin light chain, that's a proxy for an activated non-muscle myosin. So I'm showing you in green here that this molecule is right on top of these junctions as these cords are opening up lumens. So if you look at these little dots here, as this lumen is opening up, you can see it's activated right on top. So is actin, which binds to the non-muscle myosin. So the point is you have the whole actomyosin machinery right on top of these junctions as they're moving out of the way. So this is wonderful because we have the right players at the right place to be playing a role doing lumen opening and clearance of these junctions. So one of the next experiments we did was just to ask, okay, is the cytoskeleton and is non-muscle myosin activity really important for this opening of lumens? And so this is just an experiment using cytoskeleton, which is sort of a sledgehammer against actomyosin, against actin, and therefore actomyosin. But when you open up these lumens, what happens is if you treat them with cytoskeleton, then the lumens do not open. And this is a system in which we have live embryos in rolling bottles, and we simply add, reagents, pharmacological reagents, as they're opening up these lumens. We then did an experiment to say, okay, beyond the actin cytoskeleton, what about the actomyosin contractility machinery directly? There's a drug that you can use to get to that question, and that's blebistatin. And blebistatin will actually inactivate non-muscle myosin. So with both of these experiments, it's kind of a knockout experiment. And we asked the same question, does it affect formation of these lumens? And you can see nice open lumens here in the dorsal aortae, nice open lumen. And here's what happens when you affect either the F-actin or either the activity of non-muscle myosin, and it keeps them closed. Yes?
Is the actin anchored to the adhesive domains like for focal adhesion?
I should have covered that, but yes, absolutely. So these adhesions are anchored by the cytoskeleton. Left out that very important slide, you remind me. Okay, so players are in the right place at the right time, and they are critical for it. So can we show that RAS-IP1 is really important for it? And here's that experiment here. Normally, in a RAS-IP1 head, for instance, here is the active non-muscle myosin, and here's the junctions right on top of each other. And here's what that myosin looks like, that phosphorylated myosin, in the absence of RAS-IP1, critically required for the activity of myosin light chain. So I'm going to sort of summarize a lot of work, but we were able to place RAS-IP1 sort of ahead of this actomyosin contractility, phosphorylated myosin light chain and non-muscle myosin, and place that ahead of this apical clearance and lumen formation, which was really wonderful. However, there's a lot of business being done between RAS-IP1 and between this non-muscle myosin, and that includes a huge number of kinases and GTPases that are in this area right here. And I'm going to summarize this, but the work ended up being sort of cracked open when I collaborated with this guy, George Davis, who at the time was at A&M. And he had spent basically all of his career looking at lumen formation by endothelial cells in vitro, and I was doing it in vivo. And so I said, hey, what about taking a look in your system? Could you see what happens when RAS-IP1 is gone? Are lumens affected when that happens? And take a look at these GTPases. He was a real big GTPase guy. And what he saw was that when either RAS-IP1 or this RGAP29 were removed, when we did siRNA, active CDC42, which is one of these GTPases, active RAC, the activity would just crash completely. So you can see the bands are missing here, but not the total. So those proteins are there, they're just not activated. So somehow RAS-IP1 is really critical for the activity of these GTPases. Rho, on the other hand, was doing something different, and it was going up instead. And so the bottom line of these experiments was that we could say RAS-IP1 is really critical for CDC42 and RAC, but it also suppresses Rho activity. And that results in the proper regulation of the actomyosin contractility and the clearance of these adhesions. So I'm just going to sort of summarize all of this work. It's a lot more complicated. It involves a lot of knockouts, genetic knockouts in mice, a lot of pharmacological knockouts. But the fun thing for this particular talk, I think, is just to understand the process of how a molecule like RAS-IP1, a boring old scaffolding protein that is just bringing together other catalytic molecules, such as these GTPases, how it can be really important for the morphogenesis of these blood vessels. So these angioblasts come together, they make junctions. These junctions must be moved out of the way so that now, as the heart starts to pump, fluid starts to be put into these lumens. It now forces open these lumens. RAS-IP1 was really critical, and I'm not showing it to you in this talk, but for activation of CDC42 and for that movement of these junctions out of the way. Now, the interesting thing about development is that it's a really three-dimensional process, and you were asking, is it the same anterior, posterior? And it's not. And what we know is when blood starts to pump in, it pumps in and it puts pressure on endothelial cells. And by a different mechanism, RAS-IP1 via RGAP29 influences a different set of GTPases, including Rho, that are required for relaxing the contractility within these cells and allowing them to become very thin, go from very cuboidal to very thin, and open up that lumen. Okay, so it's a lot of work really quickly, but I want you to remember about RAS-IP1. I want you to remember that it is required for the activity of these GTPases to change cell shape and to change junctions. Yes, please. And I can take water because I'm just talking.
It's directional. What's controlling the direction of presuming it precedes the flow?
Really great question, and I'm hiding 2 or 3 slides that explain that. I was wondering whether anybody would ask. It's actually not directional. So, can I unhide? I don't know how to do that. I'm not technologically savvy enough. But let me just exit this because I think it is actually a really important question. Yeah, there we go. Let me see if I can jump on here and show this slide here. There we go. Okay. When you take a cross section through a tube, the junctions look like points, right? These little point junctions, they look like just a single little point. However, what they are really is they're actually long ribbons, okay, of junctions. And I'll show you that. Actually, I can just show, it won't activate. Let me see if I can activate it. There's just a part that I need to remove here. A black box that need to move out of the way so I can actually show you that. Okay, I can show hidden slides. Okay, so these junctions are actually these long ribbons when you go up and down the tube that have to move out of the way. So how is that happening? In cross section, it looks like these little individual points, which the arrowheads are pointing to. But now if you look at en face, you can see these long ribbons. So how do you move them out of the way? Contractility is what is shrinking these. So over time, as you're opening up these lumens, all of these ribbons are shrinking, and they're actually shrinking to tricellular junctions at the periphery of this tube. So you can see that all of these ribbons are clearing from this. So it's not actually directional. It's shortening of these junctions that's happening through this contractility. Thanks for asking it. I wasn't going to go into it, but yeah, please.
So I'm just wondering if there's a role for RAS-IP after luminal expansion, because in your matrigel and frog experiments, it looks like after the vessels are formed, if you knock it down or inhibit it, there's a.
Neither one of these experiments address after.
Okay.
So, but we did address after in a series of other experiments, doing a conditional knockout in mouse. And they're very sensitive to absence of RAS-IP1 early, less sensitive. We knock it out in adults and they're still alive. Now, we have done matrigel implants, so an angiogenesis kind of cancer assay, and it does inhibit angiogenesis, but it's always growth of new blood vessels. Even though RAS-IP1 is present in adult blood vessels. So we don't know if it's there to respond to stress conditions, but at least in, just normal conditions, it doesn't seem to affect. Great question. Thank you. Yes.
It's a apical basal polarity in the endothelial cells during this process. So are the ZO1 adhesion points maintained as apical entities in an apical basal polarity, and so they're therefore
Also great question. And it's why epithelial people hate endothelial cells, because ZO1 is not actually apical in endothelial cells, because there's an apical and there's a basal, but on the lateral sides, there is no such apical basal region. There's just a point, right? So yeah, we are curious about that too, about the analogies between, lumen formation, but ZO1 is just not apical for us. It's just junctional.
the pancreatic example.
Right, which we need to move to.
That's the.
And here's what I was skipping about the does it all happen at once, lumen opening? This is a catching through an early embryo where this is where the heart is. You can see open lumens up here, and then you can see back here it's closed lumens, and even back here it's only angioblasts. So it's an amazing, that's why development is so fun, is that it's so three-dimensional, four-dimensional, even with time. So anyway, okay, let me move on. So the main point so far, and oh my goodness, did I go that slow? I'll try to speed up, is that it's required for GTPase activity and it controls lumen formation. So the great question that my chair asked me during a faculty meeting was, is there such a control node, such as RAS-IP1 in other systems like epithelial systems? And I told him, no, there is not. And then I thought, after I left my faculty meeting, I thought I should really check that out. And I was wrong. So it turned out that there is another molecule called ephadin that's very similar, in fact, to RAS-IP1. And what I'm going to show you in this next part is that it is also a GTPase controller. It is a scaffold protein that is also really important for GTPase activity, but also for lumen formation. So I show this picture again. This is the plexus that I mentioned. This is the system of tubes that was discovered by Ali, who was one of my first graduate students. And she came in and we had totally different projects in the lab about pancreases. And she told me, she said, in these particular mutants, she said, the branching looks different. And I said, oh, you can't say that because nobody's looked at branching in pancreas. And she said, well, I'm going to look at it. And I said, okay, well, and at the time we were thinking that we were going to establish the way the pancreas branched. And there was a beautiful paper that had just came out in Lung by Mark Krasnow, showing that it was very stereotyped. Every branch, you could tell where it was going to be when, and we thought we were going to do that in pancreas. But as it turns out, pancreas develops by a completely different mechanism, which is wild. And this is what we discovered. If you look at the little branch, little buds on the pancreas and how it grows, what we noticed is that, they didn't do what we expected. We expected them to grow out and then to branch again, because that's the way the lung does it. That's the way mammary glands do it. That's the way the kidney does it. But instead, what we noticed, if you notice these brackets, we noticed these little bumps were always the same size, except there was just always more of these little bumps. And we're like, why is that happening? The tips are not lengthening. but they're increasing in number over time. So we decided we had to this was a look from the outside. This is the epithelium you're looking at. This is a LAC-Z reporter that's expressed throughout the epithelium. But we thought, okay, well, let's look at the lumens. What are the lumens doing during all of this? And this is what she saw. So let's actually start off at the right-hand side. The pancreas is like broccoli. It's very dense branching structure. And it has this plexus, like I mentioned, which actually resolves at birth. But during all of development, it has this plexus. And if you start going back in time in development, the plexus is always there. And I was like, well, let's find the beginning of this plexus. When does this emerge? And if you go back all the way to the very beginning, when the pancreas is just a placode, just a little bud, what you find is that it's smooth on the outside. It has no hints of branching. It's like our mind couldn't even understand this. But if you look on the inside, it's filled with these lumens that are, all branched already. So it's branched on the inside, but it's not branched on the outside. So this was, just really fun and new biology. So we and others all sort of came onto this around the same time. And I really like this representation. So let me show you. Early on, the pancreas comes from the gut tube, and it's going to branch out like a tree, but it doesn't do it the way we expected. And what we found out was that it forms this initial little placode that is the stratified epithelium. The cells just kind of all pile up on top of each other. And then what they do is that they create this bag of epithelium that's like thick and it's piled up on top of each other. And then at some point, it undergoes this change where within these stratified epithelial cells, they start to do weird things. They start to change shape and they start to rearrange their junctions. And to our eye, it was like, oh my God, that's what the blood vessels are doing, except kind of different. And so we got really interested in how junctions were moving around and how apical basal was changing. And then in the end, these little microlumens here all reach out, they touch each other, they form a plexus. This plexus is very complicated, three-dimensional, like I showed you, although it eventually resolves in time. And then it's like a tree that looks just like the mammary tree or the lung, which is just simple cuboidal epithelium. So our lab is interested in how that happens, where the progenitors are, where the islet cells early, and none of that is known. But just to show you this early work, Allie was furiously just trying to understand this. And so this is a cartoon we made to represent what all of her stains were looking like. But she found that, in this bag of epithelium on the outside, there were these semi-polarized cells that had a basement membrane. So they kind of had basal, but they had no apical markers whatsoever. And on the very inside, this is the lumen that's attached to the gut tube where the placode is growing out. They had apical, but they didn't have basal. And then they had all these intervening cells that were completely apolar. And these cells were doing really cool things because if you looked, for instance, at this Z01, which you just mentioned, Dr. Lamantia, that should be apical, you would have a cell that would have this apical marker on all sides of it. But then these cells would start to change shape. They would start to undergo this bottle cell formation. They would start to aggregate the Z01 in one spot. They would form these little rosettes. So all these cells would say, okay, let's all constrict our apical sites coordinately. And then in the middle, we'll start building a lumen. And then they start building this little microlumen there. And then they put these Z01 molecules at the right place. So this to us was amazingly similar, even though different than the blood vessels. So we started thinking, could there be, my reading informed me that in fact there was kind of a RAS-IP1 in epithelium called a Fadden. It was a little bit different. It had some of the same domains, although it had a very clear actin binding domain. And the wonderful thing about a Fadden is that there was a lot known about it. And both of these molecules are related to each other. They have an ancestor molecule called canoe in Drosophila. So with canoe and with a Fadden, I could learn what was in the literature about them. Turns out a Fadden binds to some cell-cell junction molecule, adhesion molecules, and it also binds to the actin cytoskeleton. And what it does is that it actually catalyzes maturation of junctions. Nectins, which are the very first two cells moving around, they meet each other. First thing they do is put nectins down. But they will strengthen junctions by recruiting things like cadherins. And it turns out that a Fadden is really at the nexus of all those decision-making processes, and it's controlling via these GTPases. And I was like, I'm starting to think that there's similarities between the epithelial and endothelial cells. So what we decided to do is to knock it out in the pancreas. And so here's this pancreatic epithelium. Now here is one of these nascent lumens that I mentioned that's just kind of appearing in this big bag of epithelial cells, opening up these lumens. And ephadin is right there poised at this nascent lumen. And it's also there as the lumens start to mature. And I'm using this marker here, mucin, that is an apical marker, again, a sialomucin that coats the inside of these lumens. So you can see how that's being brought in these cells and organizing into this lumen. So Burfin, who is actually now a faculty at UNC Chapel Hill, did this work to just ask, is ephadin required within that epithelium? And just briefly to show you that it is. And so normally you should form these beautiful lumens within the pancreatic epithelium. And this is what it looks like when you knock out ephadin. And to our eye, this was very similar to the RAS-IP1 phenotype in the following way. One, there was no coordinated lumens. And two, well, I don't show you there. Okay, two, the junctions were misplaced. Occludin is a junction that's green, and it's instead of all being where they should be to form the lumen, it was kind of all over the place. So lumens are wrong, they're not placed properly. And then also the cell shape is very different. Normally they should be more columnar, and here they're much more rounded or cuboidal. So one of the things that ended up, and I know I need to go fast, but one of the things that ended up being really clear during her experiments was that this marker that should be placed at the apical membrane was actually caught inside of these epithelial cells. Now, this was something that was not really possible to observe in endothelial cells, whether there was intracellular trafficking defects, because endothelial cells are so skinny. And I know you'll argue with me, but for us, we're like, we can't see anything in endothelial cells. But in the epithelial cells, they were so thick and chubby that you could really see what was going on inside. So, okay, so this is the overall phenotype that she saw. Instead of this nice plexus, all of these lumens all attached to each other in this three-dimensional sort of arrangement, you saw all of these little balls instead. So we became really interested about what is going on with these balls. Why are these things so disconnected from each other? So Burfin started having a really close look. And what I want you to see up at the top here are wild type pancreata, and down here are knockouts. And if you take a real good look, here is an epithelium with one of these lumens opening up in red. This is mucin at the surface. If you look at our mutant cells, here is a single cell surrounded by E-cadherin, so it just outlines it. And look at what's happening inside of the cell. You have this little vacuole, this little intracellular trafficking that's somehow gone awry. All the red here should be put out at the apical surface, and instead it's stuck inside the cell. Same thing, if you look for junctional molecules, it should be placed at this lumen, but instead what you're having is it's somehow stuck inside the cell. So we got really interested in whether or not perhaps ephadin was regulating some GTPases within these epithelial cells that was somehow interfering with trafficking within these cells. And so this is just a close-up of one of these things that should be getting to the apical surface. We don't know what to call them, whether they're vacuoles or vesicles or endosomes, but they're not making it. And instead, what they're doing is that they're remaining within the cell. So we turned naturally, instead of CDC42 and RAC and RO, we asked directly about GTPases that are known to be really important for vesicle trafficking, which are the RAB molecules or endosomal trafficking. And that includes GTPases such as RAB11 or 8. And what I want to show you is that we started taking a look at these GTPases, and we were struck by the fact that they were right there at the apical surface during microlumen formation. So here's mucin 1 showing you where the microlumen is, and here is RAB11 right there. But in our mutants, it was like everywhere within the cell. The subcellular localization of this GTPase was lost in the absence of the scaffolding molecule that seemed to be critical for lumen formation. Same thing with RAB11, with RAB8, and actually same thing for CDC42, which we were also shocked. CDC42 is right there at this nascent microlumen, and yet in our mutants, it's throughout the cytoplasm. So it's not the absence of these GTPases. Possibly their activity is messed up here, although we didn't test it, but their subcellular localization is screwed up. So we were really excited because there's a guy named Keith Mostov at UCSF who spent his entire career studying these GTPases and their roles in lumen formation. And he does so with MDCK cells in vitro. And he basically, showed that all of these are really critical, and his phenotypes, when he knocks them out, are really similar to ours. If you look at this apical cargo, it should be apical, is now within the cell, within the cell, within the cell for all of these GTPases, which was really fun for us to see. We have the same phenotype, but now in vitro. So a Fadden. So that's really a good question. You mean fusion between all these teeny little vesicles, endosomes? It seems that they will not fuse with the apical membrane, but they fuse with each other, and that's why they grow in size. Yeah.
Normally, there would be fusion of.
Yes, exactly.
Special.
So I'm going to close, and I'm not even going to tell you guys about the last part of my story, but I just want to say GTPases are incredibly important. We've recapitulated this phenotype by knocking out with RAB11, same exact phenotype where you don't get these lumens within the pancreas, and you do get this sort of stuck cargo within the cell. So this has been sort of a wonderful thing. We're really interested in understanding how these lumens form, both in endothelial cells, but here in epithelial cells. And it's a fascinating process and very, very strange biology. So I will end with my model for this. So what I told you in the first story with RAS-IP1 was that RAS-IP1, or no, I'm sorry. Here, this is the one for a Fadden. Sorry, let me back up. So what I've just told you in epithelium is that this molecule, ephadin, is a scaffolding molecule that's really important for the subcellular localization of these GTPases and the vectorial transport of these little vesicles in forming that apical membrane during lumen formation. It's really important for placement of these tight junctions as well. Now, what I didn't tell you about in this Affidin story is anything about actomyosin contractility. And those are programs that are going on right now in the lab. So hopefully I'll be able to tell you about that later. Very similar to this story with the RAS-IP1 story in blood vessel cells is that RAS-IP1 is also the scaffolding molecule that is important for the placement of these tight junctions and also for the activity of these GTPases. How, why? Is it because of the place they are within the cell? We don't know really yet. We do know that it affects greatly this actomyosin contractility machinery because when RAS-IP1 is gone, you get elevated Rho, decreased CDC42, and certainly misplacement of these junctions. What I didn't tell you anything about was vesicles, and my excuses is they're too thin, but maybe I need to talk to John about how to look at that more carefully, but there's still work to be done. We do notice that RAS-IP1 does co-localize with RABs, and that was actually in our initial work. If you look here, RAB5 and RAB8, they co-localize in these plated cells. But we have knocked out some of these RABs out of the vasculature, and there's no phenotype, so it's possible that it's a redundancy issue. So I will jump past a lot of work that had to do with just maintenance of blood vessels just because of time. Got too excited about the first two stories and just do my acknowledgement. I have an amazing group of people. I want to thank the RBK for the funding as well as NIH and more recently Leduc. And just thank you for your attention and all of your wonderful questions during the talk. So thanks very much.
Good talk, I enjoyed it very much. I have one question that is,
Yeah.
Your talk remind me of location, the things, in the right location. What's the driving force for, what triggers the change in activity? It's important for those scaffold proteins, but what drives them moving?
That's a really good question. And I mean, we've asked ourselves that too. We know that there's moieties, right? Farnesylation. We have not gotten into that. We're so scared of going into lipids, but I think that's the next direction. I was scared of GTPases and I did it. So maybe I'll be able to conquer lipids. I think it has to do with lipids.
Very short.
Yes, yes.
In adult organs,
Yeah.
Angiogenesis occur, and are those molecules play a role there?
Well, that's what I was mentioning before. We knocked it out in the adult. Under stable conditions, we think it's fine. When we induce angiogenesis with these little matrigel plugs, it is inhibited if RAS-IP1 is gone. So we do think so. We just haven't, we're a developmental lab, so I'm afraid of the adult. Once again, I have to conquer my fears in science and just do the experiments, but I think that's where it leads. Thank you.
Rob, I think.
Back to the question, my misdirected question about direction. Fantastic. So,
Oh, thank you.
So, one way of, getting stuck to another place is to move it in the plane of the membrane. Another way is to have stuff uniformly distributed along the membrane, but pull out the stuff that you don't want and internalize. And I'm just wondering, have you thought about, the possibility that your, myosin machinery is actually involved in internalization? Sort of,
Totally.
Right.
Yes. And I mean, that's once again, something that we could do very nicely in these epithelial cells, but we do think, so that dilute domain, which I didn't have a chance to get back to, is a myosin 5-like domain. And so we wonder whether or not it's helping determine the subcellular localization of RAS-IP1 via the cytoskeleton cargo moving protein activity of RAS-IP1. We just don't know. We need to do the domain deletions. And that's something that Natalia Mitten sent us a whole bunch of constructs.
So filtering out of the membrane, putting forward trafficking too.
Well, I mean, when we do, we have pretty good antibodies for RAS-IP1, and in plated cells, we know that it goes between the Golgi and the plasma membrane at the surface at cell-cell junctions. We know that that kind of trafficking occurs. But my collaborator, George Davis, has actually done a lot of experiments showing that it's not just localization, it also blinks in and out. And so he thinks that there's a major destruction complex, and so it's unstable. So that's another level of control, whether or not it's even present or not. So there's just, I probably could just work on this one molecule for the rest of my life. Lots to do still, but thank you for that question.
We have time for one short question. Short, Anthony?
Sure.
Short?
Cellular diversity in the pancreas. So with the initial lumen formation, we assume it's just uniform pancreatic epithelial progenitors. But is the process of cellular diversification to get beta cells and all the other ones coincident? And are you maintaining this organization in the own progenitor cells as they're either divided symmetrically or asymmetrically? Or do you have to rearrange it to get the classes?
So that's a great question. I have a friend, Anne Grappin-Baton, who's now at Dresden, and she has a biophysicist who is trying to help her work out those things. How do all those progenitors sort out in this early plexus? The one thing we do know, which is really cool, is that the progenitors for beta cells only arise and only live within that plexus. So as branches are emanating from the plexus, so there's a plexus kind of in the central domain, and then there's sort of simple branches on the outside. There are no progenitors for beta cells outside in those outside branches. They depend on something about that plexus on the inside. So, right. And so in fact, in our ephadin mutant, it locks that pancreas in sort of that conformation of the plexus. So for a longer period of time, and we get double the beta cell mass. So critical for cell fate. So microenvironment critical. Really good question. Yeah.
Great. Thanks so much. And let's take Andi one more time.
Thank you.